Assalamualaikum. I am Irza Umair, lecturer in the Department of Applied Emerging Health Technologies, University of Lahore. Today we are going to discuss about what is centrifugation, what is centrifugation machine, its types, and how it works. Centrifugation method. Centrifugation method is basically a process of separating different components or suspended particles from the homogeneous solution by the practice of centrifugal force. Particles are separated on the basis of the properties, electrochemical properties, and physical properties. If we can look at the physical properties, what we have to consider, we have to consider the density, size, and shape. On the other side, if we can see the chemical properties, then we have to say its molecular weight and viscosity. Centrifugation method. A natural gravitation force can separate the particles, but it is a time taking. So, to increase the efficiency, a centrifugal force functions to separate the particles by their sedimentation properties. The rate of sedimentation can be increased or decreased as per increase or decrease in the gravitational force and the viscosity of the medium. What is centrifugation? A technique used to accelerate the sedimentation process by the applying centrifugal force. Different types of centrifugation techniques are used to separate suspended particles from a liquid in which particles are not soluble. Liquids of different density can also be separated out. Applications of centrifugation in separation of serum, plasma, RBCs, sedimentation of cell, viruses, separation of subcellular organelles, isolation of macromolecules like lipids, proteins, RNA, and DNA. Basic principle of centrifugation. A centrifuge uses centrifugal force to isolate the particles from their surrounding medium. When the rotor rotates at a certain speed, then the force that causes the particles to move radially outward is a centrifugal force, while the force which acts on the particle is the relative centrifugal force. Basic principle of sedimentation. When we design the centrifugal protocol, it is important to keep in mind that the denser a biological structure, the faster it sediments in a centrifugal force. The more massive the biological particle, it faster it moves in a centrifugal force. The denser the biological buffer system is, slower the particle will move in a centrifugal force. What we can conclude from these 3 points, we can conclude that the particles are denser, massive, then they are separated or they can be separated quickly because the denser particles settle down quickly at the bottom. While if the medium is denser or viscous, then it takes time to separate the particles. Then we have to see the forces that act on it. If we consider the frictional coefficient, if the force of friction is greater, the slower a particle will move. And the greater the centrifugal force, the faster the particle will move. But in case when the density of both medium and the particles are equal, then the rate of sedimentation is 0. Which factor has a greater influence on the centrifugation? Density of both sample and solution, temperature, distance of particle, rotation speed. Type of centrifugation. Based on the type of separation molecules, there are 2 significant types of centrifugation: preparative centrifugation and analytical centrifugation. Their names are showing that what is preparative centrifugation. It is a techniques for concentrating one component from the other. Means it is only a separation technique. But if we can look at the analytical centrifugation, analytical is like something to analyze. So it is a method to measure the physical, chemical, hydrodynamic properties of the suspended molecules. Principle of centrifugation method. A centrifugation method depends on the gravitational movement of suspended particles, which causes the deposition of high density particles towards the bottom. The greater the difference, the easily can the components can be separated out. But if the difference is less or low, then the separation becomes tough and it becomes time consuming. And we need greater force to accelerate the particles. Relative centrifugal force. The relative centrifugal force is a radially force generated by spinning of rotor as expressed in the relative to the Earth's gravitational force. To convert the revolution per minute into the relative centrifugal force, we can follow the following formula like RCF is equal to RPM square into 1.118 into 10 raised to the power minus 5 multiplied by R. RPM is the speed of the rotor, small r is the radius. It means Relative centrifugal force depends on two things, that is RPM and the radius. Interacting forces. Other than the centrifugal forces, there are two other type of forces that can act on the particle while the process of centrifugation. That is frictional force and the Buoyant force. What is Buoyant force? A force with which the particles must displace the liquid medium into which they sediment. The opposing Buoyant force is smaller for the dense particles and can be expressed as equal to rho gV. Rho is the density of the fluid, g is the gravitation or centrifugal acceleration, v is the volume of the fluid displaced. Frictional force, a force generated by the particles as they migrated through the solution, where F is the frictional coefficient, v is the sedimentation velocity. Parts of centrifugation machine. Basic parts are chamber, rotor, cups, motor control assembly, power switch, timer, tachometer. These diagrams show the basic parts, like that is there is a chamber, door, emergency door, open hole, control panel. The control panel, basically what we set is the time for centrifugation and the RPM value. The chamber consists of the rotor and the tubes. Rotor is a part which causes a machine to rotate and on which the tubes are placed for the suppression. Types of centrifugation is a desktop, high speed, ultra centrifugation. Ultra centrifugation has two types, that is analytical and apparatus. Type of the centrifugation depend upon the maximum speed of sedimentation, presence, absence of vacuum, temperature control, volume and sample, and the capacity of the centrifuge tubes. Types of rotor. Basically, we have three types of rotor, that is vertical tube rotor, swing bucket rotor, and fixed angle rotor. Fixed angle rotor. In this, the tubes are held at a fixed angle, that is 14 to 40 degrees. Particles can move radially outward, they travel a short distance, and reorientation of tube can occur during acceleration and deceleration of the rotor. Swing bucket rotor. Swing out of horizontally position when rotor accelerates. Longer the distance traveled, allow the better separation on the basis of the density gradient. What is the advantage of swing bucket rotor is that easy to withdraw supernant without disturbing the pallet. Normally used for a density gradient centrifugation. Types of centrifugation is small bench, micro centrifuge, high speed, ultra centrifuge, large capacity, preparative centrifuge. So first of all, we discuss about the small bench top. And its name is showing that small bench top, it means it is used for the small amount of material that can rapidly or easily sediment, like yeast cells, erythrocytes. Its speed is slow, like about 4,000 RPM, common in clinical lab laboratory, blood, plasma, serum suppression, can take approximately 100 tubes depending on diameter. Where it is used? If we can see the research application, then it is used in cellular biology, microbiology, genomics, molecular biology, biochemistry, pharmaceutical studies. And clinical applications, clinical chemistry, clinical microbiology, hematology, clinical studies. Microcentrifuge means it takes a small volume, very common in biochemistry molecular labs, and it can generate the force up to 15,000 G with or without the refrigeration. High-speed centrifugation. What is high-speed centrifugation? Its name is showing that it works at a high speed. Its speed is approximately 15,000 to 2,000 RPM, and centrifugal field varies up to 1 lakh. Large sample capacity depending upon rotor, research application, different separation of nucleus, mitochondria, protein, and precipitates. Ultracentrifugation. Ultracentrifugation has more speed than the other. Its speed is about 65,000 RPM, but its lifetime is limited. It is expensive, requires special rotor, special care. And what is large capacity preparative centrifuge? Centrifugal field values up to 3,000 to 7,000. Efficient separation of coarse precipitate or whole cell, designed to combine dependable performance and ease of the use with advanced functionality. Large capacity centrifugation produces reproducibility, separation for high throughput, applications such as blood banking and bioprocessing. Uses. Where it is used, if we can see the environmental sciences, then the centrifugation is used in the wastewater treatment. Molecular science, it is used in the separation extraction of biomolecules like DNA, RNA, protein. Medical research in help in the separation of different components from the urine, blood, and the blood serum. Chemical sciences help in the process of uranium enrichment. Food sciences help in the production of skim milk by removing the fat. Therefore, we conclude that centrifugation techniques have a broad applicability in the field of research, biochemistry, molecular biology, cellular biology, and medical sciences. Thank you.